what we were interested in is when I first got here, I noticed last summer that there was a large fish kill on Lake Bemidji. The Tullaby and the Cisco and some white suckers during the middle of August, they die. So we know why that happens. It happens because the water, they need cold water that has a lot of oxygen in it. And what happens in Lake Bemidji is that the warm water on the top isn't cold enough for the fish to live in, but the deep water that's cold doesn't have enough oxygen. So the fish get pinched, and as a result, they die. But not all of them do. So we wanted to know what volume of water is available to these fish that has enough cold temperature for them and also enough DO or dissolved oxygen for them to survive. How much water in the lake is actually available to those fish? And then we've all heard about global warming, right? So what we're going to do then is simulate what would happen if air temperatures increase by 3 degrees like what is projected, and then what will happen to those same fish? How much will that amount of volume of water that's available to them in the peak of summer change? So that's the main problem. Is right now there's a fish kill. We know why it's happening. We want to know exactly what is going to happen if air temperatures change like is projected. So the problem is that those plants that are down at the bottom and the production that's happening in the bottom, that decomposition is too great and it consumes all that air down below at the bottom in certain lakes. And Lake Bemidji is one of those. We're going to have you bounce around and use all of these pieces of equipment and learn how to use them. See how small the mesh is on this? So when you pull this through the water, it collects all those tiny organisms, and then we can count them back in the lab. So throughout the summer, as the water conditions change, we want to know how the abundances and counts of those tiny organisms change. And we want to relate that all back to that stuff that I mentioned earlier about how there's no oxygen at the bottom and there is oxygen on the top, but it's warm water compared to the cold water on the bottom. So this device here, which is a dredge that opens up like this, and then you drop it down to the bottom, and then it snaps shut and takes a scoop of dirt off the bottom. So that'll collect all the small insects that are living down on the bottom of the lake. So that gives us 3 different scales to measure the conditions of Lake Bemidji as it progresses through the summer. We got the macroinvertebrate scale, or the insects that are bigger. We then have the environmental condition, and then we also use that other mesh to sample the tiny organisms or animals. We also have a GPS. Does anyone know what that is? Yeah. Yep.
Where are we?
Yeah, right. So we have that device out here, a handheld GPS, just like you would have in your cars. So we can have you use that today, so we can come back and take samples at the same location again at a different point in time if we need to. And we have some labels that you can put in each one of those jars too, that has the location, the date, the time, what gear did you use, so that an Ekman dredge, and then your collector's name. So that's important, so that if the data that comes from those invertebrates or those animals we can use in the future. Okay, I think we should get into groups and grab a clipboard, and hopefully everyone has their clipboard filled out and a jar with insects in it and a jar with phytoplankton in it by the time you're done.
And a GPS.
And a GPS point.
Are we ready to drop?
Grab it. Now drop that weight. Let go. Let go.
So go.
Now we're supposed to pull it up. Okay, ready?
Ready.
No. Okay.
And then once it hits the bottom, then you go like this. Just pull it back up quick. See how everything went down into this jar? And look at all the little things swimming around in there.
Okay. We test the temperature in different deep, and our data we find the temperature. If in more deep, you will find the temperature was lower. Yes.
啊，我看到了。
Yeah, let's have
Wow.
Yeah.
This is a screen that shows you how many satellites you have. Each one of these is a satellite.
This is a word that I think you do not know.
This shows your location. Those are little organisms, microscopic. But that's what a lot of little fish eat. So it's the base of the food chain.
Okay, Todd.
You want to put them under the scope?
It's a worm.
So a baby fish likes to eat these little guys that are swimming around. And then as the fish gets bigger, it can finally switch to something like this. And then if you have a perch that's that big, it actually switches so it actually eat fish. But this stuff that you see in the screen here is the base of the food chain in that way. We also have our fish collection here, so all the fish from Minnesota. If you've never seen a gar,
It's like I took zoology.
very ancient fish.
Like a little alligator.
Yeah, little
You saw
alligator. One of
that?
them is actually called the alligator gar, right? A bowfin, found more in swampy waters.
Oh, that's an ugly one.
He's got a fake eye spot on his tail. And then little pike, like that picture of the big one that I was holding. There's a younger one. Baby sturgeon. And then here's a paddlefish, a baby paddlefish. Now these get huge. In Mississippi River and larger rivers, and they eat all those little zooplankton that are swimming around. They're filter feeders, so that's what they eat. Whereas this sturgeon is more of a bottom feeder. You can see his mouth is on the bottom, so he's eating detritus and those worms that would be on the bottom. So remember when we were out there and we took a waypoint at the location that we were at? What I did and what I want us to do is I took these waypoints and I entered them into Google Earth down here on the bottom. So you can see where on Lake Bemidji that we collected our data. Okay? Try to help answer our research question, which was how is the changing water temperatures and O2 levels in Lake Bemidji affecting the fish populations as summer goes on? And it's at two different dates, June 22nd and then a few weeks later in August 3rd. And we need to figure out here that we're going to try to make a graph that shows how the oxygen level compared to depth changed by those two dates. Okay? So, 0 is the surface, and as you go further down, this is 2 meters deep, 4 meters deep, and then this is the oxygen level. So, what we want to do is make a graph now. So we've got to go up into the top tab up here and click on Insert. And we're going to want to take that first one on the left, right there. So click on this, and then you'll click on that. And you see where it says Select Data? That's where we need to select the data to put into this graph. We want you to right-click on this figure, move up, and click on that, Select Data. Okay. What we want to put on our x-axis is going to be the actual O2 levels. And then we want this to be our depth. So now, when you look at that graph, what does it tell you? Is there more oxygen at the surface, or is there less? Top, right? Does everybody understand that? There's much more oxygen at the top, right? And fish need oxygen. So as you get deeper in the water column, the oxygen gets less and less. These are actual measurements that we took from Lake Bemidji. So you're getting down here near the bottom, and this is in June. There's only
Well, let's see, what do you?
about 4 milligrams per liter of oxygen. Fish are going to start to have a tough time down the bottom there.
Okay, hang on.
So our next question is, how does this pattern, the amount of oxygen at different depths, change as the summer progresses, right? That's why I have 2 dates here. So here's June 21. We need to make the same figure for August 3rd, but you've already done it once. So all you need to do now is right-click on that figure, copy it,
Huh?
paste it right below, and when you click on that data, you can see where it is. All I have to do is drag that down, and it'll change the data. You see how the lake becomes stratified, where there's a lot of oxygen on the top, and then very little on the bottom? What do you think is harder on the fish? Is it harder for them
Yeah, let's just see.
in August, when it looks like this, or is it harder for them in June when the figure is like this? August, right? It's much worse in August because the whole bottom half of the lake has less than 2 milligrams per liter of oxygen. So basically, once you get below 6 meters, 7, 8 meters, the fish can't even utilize that habitat. So all I did was copy that other figure right into this next tab over, which is the temperature tab. So that one is for June and one is for August. And there, I have both my temperature graphs made now. All you need is 2 figures again, right? And one is for the temp data in August and one is for in June. Okay, so what is that? And this is from June, okay? So right here, this is the cutoff, right? Ciscos can't live in water that's over 20 degrees. So anything that's blue up on the surface, like a light blue like that, they can't live in. So this section in June is too warm for them. Now we should be able to go through these fairly quickly here, because all you have to do is go down and highlight the next section.
So go back to the Word.
Yep. Copy it from the Word document.
Copy back to R.
Go back to R, paste it in, and hit enter, and then you'll change, and it'll be the DO graph, or the O2 graph. The fish can only live in the middle during June, right? Why can't they live up top? It's not cold enough, right? It's too hot. It's over 20 degrees. And why can't they live down the bottom? Yep. There's no oxygen. It's below 3 milligrams per liter. So by combining those 2 maps, we're able to show the actual amount of water, or the volume of water that a fish would be able to live in the North Basin in this July. Okay, so this is our surface. This is down deep. The fish can only live in the middle during June, right? Why can't they live up top? It's not cold enough, right? It's too hot. It's over 20 degrees. And why can't they live down the bottom? Yep. There's no oxygen. It's below 3 milligrams per liter. So by combining those 2 maps, we're able to show the actual amount of water, or the volume of water that a fish would be able to live in the North Basin in this July. Okay, so now let's do the same thing for August. Copy and paste it, and you should get a figure like this, and that shows what habitat is available to them in August. And interestingly, there's some really weird things that happen. The top is available to them in August. Now, we had such a strange summer that it was actually cooler in August than it was in July. And normally, that's why we didn't have a fish kill this year, because they can still have access to that upper water column.
So for many, many years, every August, the fish die.
Oh.
But this year, the fish do not die.
Yeah.
So you collected data on the lake, and Andy collected data, and the data show that, yeah, the fish should not die this year. Because the temperature was cool enough. Huh.
So that makes you guess. That makes you think now. The next hypothesis, and the reason this is important, is what happens if we have a climate change scenario where the temperature increases. So the next one down, you'll notice, if we go to the third one, on the next scenario on the Word document, it's a climate change scenario, and I increase the temperatures by 1 degree. Okay? So let's do that. Let's copy and paste that in and see what happened. What's the big thing you noticed?
Oh, geez.
What happened?
Space is small.
Too small.
Very small.
Yeah.
So just by raising the water temperatures 1 degree in August, you would limit the amount of area or volume up to those fish to an enormous amount.
Wow.
So imagine under a climate change scenario where it's going to range, 3 degrees on average. Those fish are going to be in big trouble, aren't they? So that's the big picture of this type of research, and why we're interested in it is I can now project what can happen in Lake Bemidji under different climate change scenarios, just from that data that we collected.